Okay, it's time to get on with the second talk in the series of 3 on protein interaction networks. And so this time we have the dimension of protein structure being introduced into the networks. And so it's a pleasure to introduce Brendan Jha from Boston University. Thank you.
Okay, so thank you very much for the nice introduction, and I want to thank the organizers for giving me the opportunity to present our recent work on structural modeling, three-dimensional structural modeling of protein networks between species. So the theme of this session is a protein-protein interaction network. So this is, you have seen this a million times, and then each node is a protein, each edge is a protein-protein interaction. What we're interested in is to produce a more high-resolution models of these nodes and edges. In particular, we want to produce atomic level resolution models of nodes as a protein structure, as well as structural models for protein-protein interactions as interacting proteins sharing a particular interface. So there are two questions. One is this feasible to do? And the second question is, if it's feasible, is it useful? Does it provide any additional insights that goes beyond the traditional binary protein-protein interaction network? So for the first question, we know that it's very difficult to de novo predict protein structures as well as structure of protein-protein interactions from first principles. But for a subset of these proteins with known structural homologs, we can use homology modeling. And homology modeling has been surprisingly effective these days. So the idea of homology modeling is quite simple. So we start with a query sequence. We assemble a library of protein structures that have solved crystal structure in the protein data bank, and we do similarity search. We find the best match if we're lucky, and then we use this best match, a significant best match, use this structure as a template to construct a structural model for the query sequence. So it turns out that homology modeling is surprisingly effective. Based on the estimates both from databases such as PFAM and Superfamily, more than half of all proteins share significant sequence similarity with at least one structure template protein in the protein data bank. And that's pretty good. Moreover, if for your protein of interest, you can find significant sequence similarity with a template protein with a known 3D structure, then with very high probability, the two proteins will share significant structural similarity, and this is known ever since the pioneering work of Childe and Lesk back in the 80s. And this tells you that it is, that you can construct a good homology model using this template protein as a structural template. Moreover, with reasonably high probability, you can identify the correct sequence alignment between the two, and you can construct a good homology model based on these. So the end result is for any new protein of interest to you, one can construct an accurate homology model for more than half of the time. Now, the caveat of this is, this is only promised for a part of the protein. This may be a big part, but most likely it's a small part of the protein. It may be a protein, it may be a part of that you're interested in, but it also could be a part that you're not interested in. So, but with that caveat in mind, still I think it's pretty good that homology modeling, one can apply homology modeling on a genome scale with reasonable success. And the same thing is true for homology modeling of protein-protein interactions. So given a new pair of protein-protein which are known to interact by independent experiments, for example, by is2 hybrid or pull-down experiment or any other classical biochemical experiment, then you can go to the library of protein-protein complex structures in the protein data bank and do sequence similarity search. And if you're lucky, you find a significant match. You use that matched complex structure as a template to build a homology model for your querying protein-protein interaction pair. It turns out that you can also do this. You can build accurate homology models for a small fraction, but still significant fraction, roughly 20% of known protein-protein interactions, depending on how you define as known protein-protein interactions. So the bottom line is it's feasible. It's possible to do this. Now, the big caveat is we can only, so here's a picture showing this is the binary protein-protein interaction network. And here, the green edges are the structural, the part of the protein-protein interaction network with a structural model. So it's a small subset. It's not an unbiased subset. It's a biased subset. For example, it is depleted of membrane proteins, depleted of disordered proteins. It's enriched for stable rock-like proteins, highly expressed proteins, so on and so forth. But with those caveats in mind, there's a lot of good things that come with these structural interaction network. In particular, a lot of many presenters in this conference have talked about, have given great examples of the utility of structural interaction networks. So within the context of this particular talk, I just want to highlight two particular properties. One property is with the knowledge of a three-dimensional structural model, one can start to think about geometric relationships between different edges. So here, the edge is actually interface. So if you just have the binary network, you don't know. You just know that these two proteins, this hub protein talks to these two proteins, but you don't know whether or not it talks to these two proteins using the same interface or using different interface. But with structurally resolved models, you can start to think about geometric relationships between these interfaces, and this will become important in a moment. And the other thing we can do is we can construct very high-resolution models relating the biophysical properties of proteins to evolutionary properties of proteins at very high resolution, in fact, at a single residue evolution. So here's a picture showing you here is the interaction site. It's a residue, so we can actually model, take this residue, we know what fraction of this residue surface is involved in interface, participation involving protein-protein interaction, what fraction of this is exposed, what fraction is buried. We can also quantify its evolutionary properties and build models which relate the evolutionary properties of these sites back to biophysical properties and microenvironments of these sites. And this is also very important. So you can do this not just for one protein, but across the entire proteome. And I think that's a very powerful thing to have. So typically, we think about protein-protein interaction networks. We think about protein-protein interaction networks within a particular organism, for example, within yeast Cerevisiae or within human. So we can construct structural interaction networks for these binary networks. But today, I would like to focus on a very interesting type of protein networks, which is the binary protein-protein interaction between two different species, in particular between the human protein and the virus protein. So we can also think about constructing three-dimensional models, structural interaction networks for these type of networks. So the work I'm describing here is joint with Eric Franzosa in the bioinformatics program at Boston University. So from now, we're going to refer to the human-human protein-protein interactions as endogenous interactions, and human virus protein-protein interactions, these between the blue and the red guys, as exogenous interactions. So there has been a lot of pioneering work quantifying the binary exogenous network from the work of Woods and Calderwood and Dyer, among others. So there has been a lot of impressive results coming out of the binary protein-protein interaction networks. So what we're interested in to see here is whether or not a structurally resolved protein-protein interaction network can give us any additional insights. So we are able to do this. So here's the structural interaction network that we're able to construct. So each red node here is a virus protein. Each red edge is an exogenous interaction between a virus protein and its human target protein. The blue edges are the other endogenous interactions between the human proteins involving a virus target protein, and the gray are all the other endogenous human protein-protein interactions. So importantly, each one of these nodes is structurally resolved. So it has a three-dimensional structure associated with it. Each one of these edges also has a three-dimensional structure associated with it, a model associated with it. In total, we're able to construct structural models for a little bit over 50 human virus protein-protein interactions and a little bit over 3,000 human protein-protein interactions. So what we're going to do is actually to put these two different types of networks together, the exogenous network and the endogenous network, and to compare them within this framework of structure interaction networks. So the first question we can ask is, so now, if you recall, because these are structurally resolved interactions, we actually know the geometric relationships between different edges acting on a human protein. So here's a case of a human protein targeted by a virus, but at the same time, this human protein also interacts with other human protein. So the first question you can ask is there any relationship, geometric relationship between these exogenous interfaces painted in red and the endogenous interfaces painted in blue? Are they all over the place, or do they tend to strongly overlap? So you can do these statistics. It turns out, so here's a picture showing this big gray blob is the human protein targeted by a virus. And there are two different types of interfaces. The red interface is the part of the surface of the human protein targeted by virus. This is exogenous interface. The blue interface are the endogenous interfaces targeted by other human proteins. And then you can start to calculate, for example, the degree of overlap between these two types of interfaces and plot those statistics. So here is a triangle plot on top. These are cases where there's perfect overlap. The endogenous and the exogenous interface complete overlaps. There are cases of that. On the bottom are basically no overlap. That's on the bottom line. But basically, most of the time, we find that there is a strong degree of overlap between exogenous interfaces with endogenous interfaces. And bear in mind that expectation here is very low because the protein is big. Okay? So if you just pick two random interfaces on the surface of a protein, most likely they will not overlap at all. So the fact that they overlap in any degree is actually quite significant. You can do statistical calculations. This is extremely significant. So the other thing you'll notice is that there is a general shift towards the right of the graph. So the midline here is the line of equal area between endogenous and exogenous. Going in right is where you have smaller exogenous interfaces. Going left is you have smaller endogenous interfaces. So what you're seeing here is a tendency for virus to have smaller interfaces, exogenous interfaces compared to human proteins. So here's a diagram showing that. There's a strong overlap, extensive overlap between the viral target interface with the endogenous interface. At the same time, the viral interface tend to be smaller in area. So here are a few examples. So here's a human protein CDK6. It has two binding interfaces. This one is an endogenous only interface, binds to a CDK inhibitor. This one is targeted by both an endogenous protein interaction partner, human D-type cyclin, as well as an exogenous virus protein. This one is cyclin homologue. In this very case, these two proteins are homologs of each other. Here's another case where this protein in the human binds to an endogenous partner, this blue protein. It also binds to a virus protein, but these two proteins look completely different from each other. So you can ask the question, what's the mechanism associated with interface mimicry? So we know that viruses tend to bind to existing known interfaces, endogenous interfaces, by this phenomenon, interface mimicry. But how does it achieve this? Does it achieve this by having a protein which looks similar to a human protein by means of either similar in terms of sequence or similar in terms of structure? It turns out that that's only a minority. About 30% of the cases, interface mimicry. So for example, here there's a virus mimicking a human protein in terms of mimicking this particular interface. About 30% of the case can be explained by sequence and structural similarity. But the question is this big or small? Well, you have to compare this number, 30%, to the corresponding reference state. The reference state is what happens in human. Interface mimicry also happens in human. You find a lot of cases where two human proteins bind to the same interface of a third protein. In those cases, the vast majority of these two human proteins are duplicates of each other. They're the results of divergent evolution, sharing both sequence and structural similarity, and that's the majority of the case, 80%. So there is a significant difference. Interphase mimicry by virus proteins tend not to be explained by a sequence or structural similarity. The other thing you can look at is virus targets certain interfaces, but there are many other endogenous interfaces. Why do viruses only target this particular human endogenous interface, not the myriad of other interfaces? So we can look at several properties. So for example, one property is co-expression pattern. So for example, here, this virus targets this human interface. While this interface is also targeted by many other human proteins, what about the co-expression pattern between these human proteins? Turns out that none of these cases, none of the virus target interfaces share any degree of co-expression. So it's zero. The number of endogenous protein pairs that particular interface targeted by a virus protein, none of them are co-expressed. They're all transient. Whereas a significant part of the human endogenous protein-protein interactions are co-expressed. So what this means is that virus-targeted interfaces tend to be transient interfaces. So here I put the dashed line here to represent the transient interface, whereas compared to human, a lot of the human interfaces are not transient. They're stable interfaces. So the other question we can ask is, what about other properties? So the other we can ask is, this is the virus target interface. We can count the number of other human proteins which target this particular interface. So in this case, for example, there is one here, whereas we can compare this to the interface-specific degree for other human interfaces. In this case, there's two, there are two, there are two. So we can do this calculation. It turns out that virus-targeted interfaces have a significantly higher endogenous degree compared to purely human endogenous interfaces. So here's an example where this virus-targeted interface has 10. It's interacting with 10 other human proteins, where this is exceedingly rare for the endogenous interactome. So what this means is endogenous interfaces mimicked by virus protein tend to participate in multiple human-human endogenous interactions. And here's a picture that here's a virus-targeted interface tend to interact with a lot more other human proteins compared to other human endogenous interfaces. So the other thing we can check is, what about the function of these human proteins which are targeted, which share the same interface as the virus targets. And it turns out that they're significantly more likely to be enriched for regulatory activities. So here's more than half of them are enriched. More than half of these proteins have a regulatory activity and annotation, whereas less than half, slightly less than half, in general in the human proteome have a regulatory activity. So, and here's a picture. These arrows represent regulatory activity. So it seems that there is a big difference between the mimic interfaces and other non-mimicked human endogenous interfaces. So the mimic interfaces tend to involve in regulatory activities, tend to be transient binding, and tend to participate in multiple endogenous interactions. So what this tells us is that, again, if you think about viruses being obligate intracellular parasite, there's a tremendous selective pressure. And plus, it doesn't have a lot of resources. Unlike the human genome, which is big, it has a lot of protein repertoires, it can play with these protein repertoires. Viruses are extremely small. They have extremely limited resources. They have tremendous pressure to innovate, to be able to target human cells as efficiently and effectively as possible. So that explains why they can only target selected important proteins in the human cellular circuits. These tend to be high-level regulatory components, and viruses tend to target them in the most efficiently way as possible, use small interface, and so on and so forth. The last thing that we did is to look at evolutionary properties of these interfaces. So here's a picture again showing the virus-targeted proteins. This protein, this human protein has endogenous interfaces, blue. These are human protein-protein interactions, as well as the part targeted by virus. These are red exogenous interactions, and there's a significant overlap. The purple part is the overlap between the two. So then you can ask the question, which part of these interfaces evolve slow and which part of it evolve fast? It turns out that the slowest evolving part is actually all other human protein-protein interfaces. So the part which is not targeted by the virus, if human protein interacts, the interface evolves very slowly. And that we know, and the reason for this is because of a strong selective pressure to maintain the structure and function of that interface. So this part evolves very slowly, no surprise. But the real surprise is what happens for these three parts. The part of the endogenous specific, this part only binds to human protein, and this part only binds to virus protein, as well as the part that's mimicked by both. Which one of them evolves slow, which one of them evolves fast? So it turns out that this blue part, the part of the interface that only binds to human protein, other human protein, evolves relatively slowly, and that's not a surprise because, again, this part is under strong negative selection pressure to maintain endogenous protein-protein interaction. But the real surprise is the part that evolves the fastest turns out to be the purple part, the part that's been mimicked by the virus. So despite the fact that there is a strong selective pressure to maintain endogenous protein interaction function, the part that's mimicked by the virus tend to evolve the fastest. And this is consistent with this whole concept of evolutionary arms race between the virus and the human protein. Virus wants to target human protein wants to run away from the virus, and so on. So the whole, all lines of evidence supports this concept that within the human protein-protein interaction is cooperative, right? Proteins within human, they talk to each other to achieve a common goal, which is to make cells and organisms more fit. Whereas in the virus, human-virus interactions tend to be antagonistic, right? You have all these concepts of mimicry, hijacking, arms race, and a lot of these signatures shows up in a global way, statistically significant way in the structure-interaction network. So I would like to, this slide just make sure there is actually a whole wide spectrum of interspecies interactions. So perhaps there are good viruses out there, and we just don't have enough data to be able to see them. So, and I think my time is up, so I would like to stop here and acknowledge my students and colleagues in the bioinformatics program at Boston University. We have a poster, X58. You're welcome to stop by, and I'll stop here, and thank you very much.
Questions?
Quite at the beginning, when you were talking about the fact that the human-virus interactions and the amount of times they overlapped as opposed to didn't overlap, and that was significant. Did you compare that to the amount of times that just human-human proteins decide that they have an overlapping interface? Is that just the way it is? Because it's not the entire surface of a protein that things can actually interact with usually. There are parts of it which are much better for that.
Yes. So what we did is we compare, that's a very good question. So the question is, how do you know if the overlap, I think that's shown here, how do you know if this overlap is significant or not? So what we did is we take the surface, of course you have to take the surface. You can't just take the buried part. The buried part of the protein is never involved in protein-protein interaction. So you have to take the surface of a typical protein and then randomly select two interfaces with the same area, control for the area, and then ask what is the degree of overlap expected for those random cases. It turns out that when you do that, the overlap is actually highly significant. Yeah.
Hi, thank you for your interesting talk. I was just wondering, do you have enough viral sequences to see if the part of the viral structure is also like rapidly evolving in this interface, or is it just rapidly evolving in general?
Yes, that's a very good question. So almost all of the analysis done in this talk is focusing on the human part. And the reason for that is because you can compare. You can compare the viral targets with non-viral targets. So, you know, I think it'll be very interesting to look at the viral part of the story. We haven't done that, but I think that's certainly the other half of the coin. Very important.
Okay, thank you.
Yeah.
Okay, last question.
Sorry, this is a quick one. Do you ever see, do you have in your database places where multiple viral proteins interact with the same human target?
I think, I don't remember, but I think the answer is yes. But I think there are also cases where multiple virus proteins target different human proteins, but in the same pathway, for example. So both happen, yeah.
Okay, we'll stop there. Let's thank Brandon for a nice talk. Thank you.
Thank you very much.